10 diagnostic characteristics and examples, biology and 10 economic importance of glossinidae. ChatGPT. Glossinidae, characteristics, biology, and economic importance. Glossinidae, commonly known as tsetse flies, are a family of blood-feeding insects that have profound effects on both human and animal populations in certain regions. These flies are of great medical and veterinary importance due to their role as vectors of devastating diseases. Here's an in-depth exploration of the diagnostic characteristics, examples, biology, and economic importance of glossinidae. Diagnostic characteristics. Biting mouthparts. Tsetse flies have a specialized proboscis for piercing the skin and feeding on blood. Distinctive wing venation. Their wing venation pattern is unique and characteristic of the family. Vector of disease. Tsetse flies are vectors of trypanosomes, the parasites responsible for African trypanosomiasis, sleeping sickness in humans and Nagana disease in livestock. Examples of glossinidae. Glossina morsitans, found in parts of sub-Saharan Africa and a vector of both human and animal trypanosomiasis. Glossina palpalis, also found in sub-Saharan Africa and a vector of human African trypanosomiasis. Biology and ecology. Blood feeding. Tsetse flies feed exclusively on blood, and they are important vectors of trypanosome parasites. Habitat, found in various habitats, including forests, savannas, and riverine areas. Economic importance. Human health impact. Tsetse flies transmit African trypanosomiasis, a deadly disease that affects humans and causes significant morbidity and mortality. Animal health impact. Tsetse flies also transmit Nagana disease, which affects livestock, causing economic losses in agriculture. Vector-borne diseases. Human African trypanosomiasis, HAT. Tsetse flies transmit the trypanosoma parasites that cause HAT, a debilitating and potentially fatal disease. Animal African trypanosomiasis, AAT, Nagana disease, caused by the same parasites, affects livestock and impacts agricultural productivity. Healthcare costs. Treatment and control. Tsetse fly transmitted diseases require medical treatment, leading to healthcare expenses and resource allocation. Agricultural productivity. Livestock impact. Nagana disease affects livestock health and productivity, leading to economic losses in meat, milk, and labor. Zoonotic impact. Human-livestock interactions. Tsetse fly transmitted diseases affect both human and animal health, leading to complex interactions between humans, livestock, and wildlife. Livelihood impact. Impoverishment. Tsetse fly transmitted diseases can lead to reduced agricultural productivity and impoverishment of affected communities. Vector control efforts. Integrated pest management. Control of tsetse fly populations involves measures such as insecticide-treated targets, traps, and sterile insect techniques. Wildlife conservation. Wildlife health. Wildlife health. Tsetse fly transmitted diseases can also impact wildlife populations, affecting biodiversity and ecosystem dynamics. Research and disease prevention. Medical research. Tsetse flies are studied to understand the transmission dynamics of trypanosome parasites and develop control strategies. Regional impact. Geographic focus. Tsetse fly transmitted diseases are mainly found in sub-Saharan Africa, impacting health, agriculture, and development in affected regions. In conclusion, glossinidae, or tsetse flies, have significant medical and economic importance due to their role as vectors of trypanosome parasites causing African trypanosomiasis in humans and Nagana disease in livestock. Their impact on human health, animal health, and agricultural productivity underscores the need for effective control measures and research to reduce the burden of these devastating diseases. 10 diagnostic characteristics and examples, biology and 10 economic importance of Drosophilidae. ChatGPT. Drosophilidae, characteristics, biology, and economic importance. Drosophilidae, commonly known as fruit flies, are a family of small insects with a diverse range of species. While many species are harmless, some have significant economic importance in both positive and negative ways. Here's an in-depth exploration of the diagnostic characteristics, examples, biology, and economic importance of Drosophilidae. Diagnostic characteristics. Small size. Fruit flies are small insects, typically ranging from 1 to 5 millimeters in length. Red eyes. Many species of fruit flies have distinctive red or dark eyes. Wing venation. Their wings have characteristic venation patterns that aid in identification. Examples of Drosophilidae. Drosophila melanogaster, perhaps the most well-known species, extensively studied in genetics and developmental biology. Drosophila suzuki, also known as the spotted-winged Drosophila, it is a pest of soft-skinned fruits. Biology and ecology. Habitat. Fruit flies are found in various habitats, with a preference for areas where fruits and fermenting organic matter are present. Feeding habits. Adults feed on decaying fruits, while larvae feed on yeast associated with fermenting materials. Economic importance. Research model. D. melanogaster is a key model organism in genetics and developmental biology, contributing to our understanding of fundamental biological processes. Pest of agricultural crops. Certain fruit fly species, such as D. suzuki, can damage crops by laying eggs in ripe fruits, leading to economic losses. Genetics and research. Model organism. D. melanogaster's short generation time and well-studied genetics make it a valuable model organism for research in various fields. Biomedical insights. Research on fruit flies has provided insights into human health and disease, including neurodegenerative disorders. Agricultural impact. Crop damage. Fruit flies can lay eggs in a wide range of fruits, leading to economic losses in agricultural production. Biological control. Biological pest control. Some species of parasitic wasps lay their eggs inside fruit fly larvae, acting as natural predators and helping control fruit fly populations. Fermentation studies. Yeast interaction. Fruit flies play a role in the fermentation process by interacting with yeasts, which has applications in brewing and food industries. Insect ecology. Decomposition. Fruit flies contribute to nutrient cycling and decomposition by consuming decaying organic matter. Educational value. Classroom studies. Fruit flies are used in educational settings to teach genetics and basic biological concepts. Pest management. Monitoring and control. Techniques such as traps and cultural practices are used to manage fruit fly populations in agricultural settings. Biodiversity studies. Ecosystem indicators. Fruit fly diversity can provide insights into ecosystem health and dynamics. Food industry. Food quality control. Monitoring fruit fly populations helps maintain the quality of stored fruits and processed foods. In conclusion, Drosophilidae, or fruit flies, have diverse roles in research, agriculture, and ecological processes. While some species contribute to scientific discoveries and beneficial interactions, others can cause economic losses through crop damage. Their significance in genetics, agriculture, and food industries underscores the need for both research and management strategies to balance their positive and negative impacts. 10 diagnostic characteristics and examples, biology and 10 economic importance of Psychodidae. ChatGPT. Psychodidae, characteristics, biology, and economic importance. Psychodidae, commonly known as mothflies or drainflies, are a family of small insects that are often associated with aquatic habitats and unsanitary conditions. While they have limited direct economic impact, they can indicate sanitation issues and sometimes cause nuisance problems. Here's an in-depth exploration of the diagnostic characteristics, examples, biology, and economic importance of Psychodidae. Diagnostic characteristics. Small size. Mothflies are small insects, usually ranging from 1 to 5 millimeters in length. Hairy appearance. They have a densely hairy body, giving them a moth-like appearance. Humpbacked posture. Their characteristic posture with a humpbacked appearance distinguishes them. Examples of Psychodidae. Psychota spp. Also known as drain flies, they are often found in moist and decaying environments. Paracoma spp. These are commonly found in aquatic habitats like streams and rivers. Biology and ecology. Habitat. Mothflies are found in a variety of habitats, including sewage systems, drains, and damp areas. Larval development. The larvae of mothflies often develop in organic-rich environments, such as sewage and decaying matter. Economic importance. Sanitary indicator. The presence of mothflies can indicate unsanitary conditions, such as clogged drains or sewage leaks. Nuisance. In some cases, large populations of mothflies can be a nuisance, especially in indoor settings. Sanitation assessment. Hygiene indicators. Mothflies can serve as indicators of poor sanitation, prompting maintenance and cleaning efforts. Environmental health. Water quality. Mothflies in aquatic habitats can provide insights into water quality and pollution levels. Disease transmission. Limited role. While they do not play a major role, some species of mothflies have been associated with the transmission of disease pathogens in certain scenarios. Ecological role. Decomposition. Mothflies contribute to the decomposition of organic matter in aquatic and moist environments. Public health awareness. Education. The presence of mothflies can raise awareness about the importance of sanitation and hygiene. Indoor pest management. Control measures. Mothfly infestations in homes or businesses can be managed through proper sanitation and drainage maintenance. Research. Ecological studies. Mothflies are studied to understand their role in nutrient cycling and their interactions with other organisms. Pest control. Biological control. In some cases, natural predators can help control mothfly populations in outdoor environments. Water quality assessment. Indicator species. Mothflies in aquatic habitats can be used as indicators of water pollution and ecological health. In conclusion, Psychodidae, or mothflies, have limited direct economic impact but serve as indicators of sanitation issues and environmental health. Their presence can prompt sanitation efforts and awareness about the importance of cleanliness. While they do not play a significant role in disease transmission, they contribute to nutrient cycling and decomposition in aquatic and moist environments. Proper hygiene and maintenance practices can help mitigate their nuisance and potential health implications. Disease vectors in this Diptera in detail. ChatGPT. Disease vectors in the Diptera flies include a variety of species that can transmit harmful pathogens to humans and animals. These vectors are responsible for the spread of numerous diseases, some of which have significant global health implications. Here's an in-depth exploration of some important disease vectors within the Diptera. 1. Anopheles mosquitoes, Anopheles spp. Anopheles mosquitoes are known as vectors of malaria, a life-threatening disease caused by the Plasmodium parasite. Female Anopheles mosquitoes transmit the parasite when they bite an infected human and then subsequently bite a healthy individual. Malaria remains a major public health concern, particularly in tropical and subtropical regions. 2. Aedes mosquitoes, Aedes aegypti, Aedes albopictus. Aedes mosquitoes are vectors of several arboviruses, including dengue fever, transmitted by Aedes aegypti and, to a lesser extent, Aedes albopictus. Zika virus, also transmitted by Aedes aegypti and Aedes albopictus. Zika infection during pregnancy can lead to birth defects. Chikungunya, transmitted by both Aedes mosquitoes. It causes fever, joint pain, and other symptoms. 3. Tsetse flies, Glossinidae. Tsetse flies are vectors of African trypanosomiasis, sleeping sickness, and animal trypanosomiasis nagana. These diseases are caused by trypanosome parasites transmitted to humans and animals through the bites of infected tsetse flies. 4. Sandflies phlebotominae. Sandflies are vectors of several diseases, including leishmaniasis, transmitted when infected sandflies bite humans. It can cause cutaneous, mucocutaneous, and visceral forms of the disease. Bartonellosis, Arroyo fever, caused by Bartonella bacilliformis and transmitted by sandflies. It leads to fever, anemia, and other symptoms. 5. Houseflies, Musca domestica. While not the primary vectors, houseflies can carry and transmit disease-causing microorganisms, including bacteria and viruses, on their bodies and through their feeding habits. They can spread diseases such as gastrointestinal infections and foodborne illnesses. 6. Tephritid fruit flies. Some species of tephritid fruit flies can be agricultural pests, damaging fruits and vegetables. While they are not primarily disease vectors, their feeding habits can lead to economic losses in agriculture. 7. Myiasis-causing flies. Certain flies, such as botflies and screwworm flies, can cause myiasis, a condition where their larvae infest living tissue. This can lead to infections and tissue damage, although their role as disease vectors is limited. It's important to note that disease transmission by these dipteran vectors is influenced by factors such as the pathogen's life cycle, the vector's biology, and environmental conditions. Effective vector control strategies, including insecticide use, habitat modification, and public health education, play a critical role in reducing the transmission of these diseases. Effective vector control strategies, including insecticide use, habitat modification, and public health education, play a critical role in reducing the transmission of these diseases. Continuation. ChatGPT. 8. Black flies, Simuliidae. Black flies are vectors of onchocerciasis, also known as river blindness. Black flies are vectors of onchocerciasis, also known as river blindness. This disease is caused by the filarial nematode Onchocerca volvulus. Black flies transmit the parasite to humans when they bite, and the larva of the parasite can cause severe skin and eye infections. Black flies transmit the parasite to humans when they bite, and the larva of the parasite can cause severe skin and eye infections. 9. Stable flies, Stomoxys spp. Stable flies are blood-feeding insects that can transmit pathogens causing diseases in livestock, such as bovine leukosis virus and bovine anaplasmosis. These diseases have economic implications for the agricultural industry. 10. Crane flies, Tipulidae. While not significant disease vectors, some species of crane flies can transmit nematode parasites that infect turfgrass and other plants, leading to economic losses in the agriculture and horticulture sectors. 11. Biting midges, Ceratopogonidae. Biting midges are vectors of bluetongue and African horse sickness viruses, which primarily affect livestock. These diseases can lead to significant losses in the livestock industry due to decreased productivity and mortality. 12. Blowflies, Caliphoridae, and flesh flies, Sarcophagidae. These flies are attracted to decaying organic matter, including animal carcasses. While not major disease vectors, they can play a role in the mechanical transmission of pathogens when they come into contact with humans or animals after feeding on infected material. 13. Deerflies and horseflies, Tabanidae. Deerflies and horseflies are known for their painful bites. While they are primarily nuisance pests, their biting behavior can lead to localized infections at the bite site. In addition to the above examples, many other species of flies within the Diptera order have the potential to transmit diseases to humans, animals, and plants. Effective disease control strategies involve a combination of vector management, surveillance, public health education, and research to better understand the interactions between vectors, pathogens, and hosts. It's important to note that the impact of these vectors varies depending on geographic location, local ecology, and disease prevalence. Dipterans and forensic entomology. In detail, very long answer. ChatGPT. Dipterans and forensic entomology. Exploring the link. Forensic entomology is a branch of forensic science that uses insect evidence to aid in criminal investigations and legal proceedings. Dipterans, or flies, play a crucial role in forensic entomology due to their rapid development and predictable life cycles. Here's an in-depth exploration of the relationship between dipterans and forensic entomology. Asterisk 1. Role of dipterans in forensic investigations. Dipterans are often the first insects to arrive at a decomposing body due to their keen sense of smell and ability to detect odors associated with decaying organic matter. They lay eggs on or near the body, and their larvae maggots feed on the decomposing tissue, aiding in its breakdown. The presence, development stage, and species of dipteran larvae on a body can provide valuable information about the postmortem interval, PMI, which is the time that has elapsed since death. Asterisk 2. Use of dipterans in PMI estimation. Forensic entomologists examine the life stages of dipterans found on a body to estimate the time of death. The age of the larva, pupae, or adult flies can be correlated with environmental conditions and development rates. This estimation is based on the principle that insect development is influenced by temperature, humidity, and other environmental factors. Asterisk 3. Succession patterns. Dipteran succession patterns refer to the predictable sequence of fly species that colonize a body at different stages of decomposition. By studying the succession of dipteran species, entomologists can infer the postmortem interval and provide important evidence in criminal investigations. Asterisk 4. Case studies and legal significance. Numerous criminal cases have been solved with the help of dipterans. For example, the Jeffrey McDonald case involved the use of blowfly evidence to establish the time of death. Dipteran evidence can be presented in court to support legal arguments and provide critical information to judges and juries. Asterisk 5. Research and validation. Forensic entomologists conduct research to validate the accuracy of PMI estimation using dipterans under various environmental conditions. Research on dipteran behavior, development rates, and succession patterns continues to improve the accuracy of forensic investigations. Asterisk 6. Challenges and limitations. Forensic entomology relies on accurate collection and preservation of insect evidence. Incorrect collection or storage can lead to inaccurate PMI estimates. The presence of dipterans at a crime scene is influenced by various factors such as temperature, humidity, body size, and burial conditions. Asterisk 7. Ethical considerations. The use of dipterans in forensic investigations raises ethical concerns, as it involves the study of insects that feed on human remains. However, the insights gained from these studies contribute significantly to justice and truth-seeking. Asterisk 8. Education and training. Forensic entomologists undergo specialized training to accurately interpret insect evidence and provide expert testimony in court. In conclusion, dipterans, particularly flies, play a pivotal role in forensic entomology by providing crucial information about the postmortem interval, crime scene dynamics, and the circumstances surrounding a death. Their rapid life cycles and predictable behavior patterns make them valuable tools in criminal investigations, aiding in the determination of time of death and other important forensic conclusions. Dipterans and biosurgery, Drosophila as experimental model in detail. Very long answer. 2 halves. ChatGPT. Dipterans and biosurgery, advancements and applications. Dipterans, specifically the fruit fly Drosophila melanogaster, have played a significant role in advancing our understanding of genetics, development, and disease. This insect model system has also found applications in the field of biosurgery, where it contributes to various surgical and medical research advancements. Here's a detailed exploration of the use of Drosophila in biosurgery. Asterisk 1. Drosophila as a model organism. Drosophila melanogaster has a short generation time, prolific reproduction, and easily manageable genetics, making it an ideal model organism for studying various biological processes, including those relevant to biosurgery. Its small size and simplicity facilitate experimentation and allow for large-scale genetic screens. Asterisk 2. Understanding wound healing. Drosophila has been used to study wound healing processes. Drosophila has been used to study wound healing processes, including inflammation, tissue repair, and cell migration. Genetic studies in Drosophila have identified key molecules and signaling pathways involved in wound healing, providing insights into potential targets for therapeutic intervention. Asterisk 3. Cellular responses to injury. Research using Drosophila has elucidated cellular responses to injury, such as apoptosis, programmed cell death, and regeneration. Understanding these processes can inform the development of strategies to promote tissue repair in humans. Asterisk 4. Genetic manipulation and tissue regeneration. Drosophila's regenerative capabilities have been investigated, shedding light on the genetic and molecular mechanisms underlying tissue regeneration. Such insights hold promise for the development of regenerative therapies and wound healing interventions in humans. Asterisk 5. Infectious disease studies. Drosophila has been used to study infectious diseases and host pathogen interactions. These studies contribute to our understanding of immune responses, wound healing, and potential therapeutic strategies for infectious diseases. Asterisk 6. Biomedical engineering. Drosophila has been employed in biomedical engineering research, including the development of microsurgical techniques and tools for delicate procedures. Researchers use Drosophila to test and refine surgical instruments and methods. Asterisk 7. Tumor microenvironment studies. Drosophila models are used to study tumor microenvironments, angiogenesis, and metastasis. These studies provide insights into cancer biology and potential interventions. Asterisk 8. Gene therapy and drug screening. Drosophila is utilized for gene therapy studies to assess the effects of genetic interventions on wound healing and tissue repair. The fruit fly model is also used for drug screening to identify potential wound healing compounds. Asterisk 9. Neural injury and repair. Drosophila's nervous system has been studied extensively, including neural injury and regeneration. Findings from such studies inform our understanding of nerve regeneration and recovery after injury. Asterisk 10. Ethical considerations and future prospects. The use of Drosophila in biosurgery research raises ethical considerations similar to those in other experimental models. As the field advances, Drosophila's contributions to surgical techniques, wound healing strategies, and medical interventions continue to expand. In conclusion, Drosophila melanogaster, as a model organism, has made significant contributions to the field of biosurgery. Its genetic tractability, short life cycle, and relevance to human biology have enabled researchers to gain insights into wound healing, tissue regeneration. Its genetic tractability, short life cycle, and relevance to human biology have enabled researchers to gain insights into wound healing, tissue regeneration, disease mechanisms, and potential therapeutic interventions. The use of Drosophila in biosurgery research holds promise for advancing our understanding of surgical techniques, wound healing strategies, and medical interventions for human health. More points. ChatGPT. Certainly, here are additional points highlighting the use of Drosophila as an experimental model in biosurgery research. Asterisk 11. Genetic manipulation and gene expression studies. Drosophila's well-characterized genetics and the availability of genetic tools allow researchers to manipulate specific genes and study their effects on wound healing and tissue regeneration. Transgenic techniques enable the introduction or silencing of genes relevant to biosurgery research. Asterisk 12. Stem cell biology and regeneration. Drosophila has provided insights into stem cell behavior, differentiation, and their roles in regeneration. Understanding stem cell biology in Drosophila contributes to strategies for enhancing tissue repair and regeneration in humans. Asterisk 13. Microsurgery training and skill development. Drosophila models provide an effective platform for training in microsurgical techniques. Researchers and surgeons can practice delicate procedures on Drosophila, honing their skills before applying them to more complex systems. Asterisk 14. Blood coagulation and hemostasis. Drosophila has been used to study blood clotting mechanisms and hemostasis. Insights gained from these studies have implications for understanding bleeding disorders and developing treatments for wound-related bleeding. Asterisk 15. Wound closure mechanisms. Research on Drosophila's wound closure mechanisms has revealed the involvement of actin dynamics, cell adhesion, and signaling pathways. These findings contribute to the understanding of wound healing in multicellular organisms. Asterisk 16. Limb regeneration. Some species of Drosophila exhibit partial limb regeneration capabilities. Studying limb regeneration in Drosophila provides insights into regenerative potential and mechanisms. Asterisk 17. Tissue-specific responses to injury. Drosophila offers the opportunity to study tissue-specific responses to injury and identify factors that promote tissue regeneration in specific contexts. Asterisk 18. Drug delivery and targeting. Drosophila models have been used to explore drug delivery methods and the targeting of therapeutic agents to specific tissues or cell types. These studies aid in the development of localized treatments for wound healing and tissue repair. Asterisk 19. Biomechanics and tissue mechanics. Drosophila studies contribute to understanding biomechanical aspects of wound healing, tissue remodeling, and the mechanical forces that influence these processes. Asterisk 20. Translation to human health. Discoveries made using Drosophila often have parallels in human biology. Insights gained from Drosophila biosurgery research can inform the development of therapies, surgical techniques, and wound care approaches for human patients. In summary, Drosophila melanogaster serves as an exceptional experimental model for biosurgery research. Its genetic tractability, ability to replicate essential biological processes, and relevance to human health have positioned it as a powerful tool for advancing our understanding of wound healing, tissue regeneration, and various aspects of surgical interventions. The insights gained from Drosophila studies have the potential to drive innovations in surgical techniques, wound healing strategies, and medical therapies for improved patient outcomes. 10 diagnostic characteristics and examples, biology and 10 economic importance of Siphonoptera. ChatGPT. Siphonoptera, fleas. Fleas, belonging to the order Siphonoptera, are small, wingless insects known for their parasitic lifestyle and notorious for transmitting diseases. Here's a detailed exploration of their diagnostic characteristics, examples, biology, and economic importance. Diagnostic characteristics. Siphoning mouthparts. Fleas possess specialized siphoning mouthparts adapted for piercing the skin of their hosts and sucking blood. Laterally compressed body. Their flattened body shape allows them to move easily through the fur or feathers of their hosts. Strong legs. Fleas have powerful hind legs adapted for jumping long distances, aiding in their movement between hosts. Small size. Fleas are typically small, ranging from 1 to 4 millimeters in length. Modified metamorphosis. Fleas undergo complete metamorphosis with 4 life stages: egg, larva, pupa, and adult. Reduced wings. Although flightless, fleas have reduced and non-functional wings. Examples. Cat flea C. Dinocephalides felis, a common flea that infests domestic cats and dogs, as well as other mammals. Dog flea C. Dinocephalides canis, similar to the cat flea, it infests dogs and other animals. Human flea Pulex irridans primarily infests humans but can also be found on other mammals. Biology. Parasitic lifestyle. Fleas are obligate parasites that feed on the blood of their hosts. Host specificity. Different species of fleas have host preferences, but they can infest a variety of mammals and birds. Egg laying. Female fleas lay eggs on their host, but the eggs usually fall off onto the ground or bedding. Larval stage. Larval stage. Diagnostic characteristics. Siphoning mouthparts. Fleas possess specialized siphoning mouthparts adapted for piercing the skin of their hosts and sucking blood. Laterally compressed body. Their flattened body shape allows them to move easily through the fur or feathers of their hosts. Strong legs. Fleas have powerful hind legs adapted for jumping long distances, aiding in their movement between hosts. Small size. Fleas are typically small, ranging from 1 to 4 millimeters in length. Modified metamorphosis. Fleas undergo complete metamorphosis with 4 life stages: egg, larva, pupa, and adult. Reduced wings. Although flightless, fleas have reduced and non-functional wings. Examples. Cat flea C. Dinocephalides felis, a common flea that infests domestic cats and dogs, as well as other mammals. Dog flea C. Dinocephalides canis, similar to the cat flea, it infests dogs and other animals. Human flea Pulex irridans primarily infests humans but can also be found on other mammals. Biology. Parasitic lifestyle. Fleas are obligate parasites that feed on the blood of their hosts. Host specificity. Different species of fleas have host preferences, but they can infest a variety of mammals and birds. Egg laying. Female fleas lay eggs on their host, but the eggs usually fall off onto the ground or bedding. Larval stage. Larvae hatch from eggs and feed on organic matter and debris found in the environment. Pupal stage. Larvae spin silk cocoons and pupate within them. Jumping abilities. Fleas are known for their exceptional jumping abilities, often jumping up to 150 times their body length. Economic importance. Disease transmission. Fleas are vectors of various diseases, including bubonic plague transmitted by the oriental rat flea, typhus, and tapeworms. Veterinary costs. Flea infestations can cause discomfort and health issues in pets, leading to veterinary expenses. Human health impact. Flea bites can cause itching, irritation, and allergic reactions in humans. Agricultural impact. Fleas can infest livestock, causing discomfort, reduced productivity, and economic losses. Pest control industry. The need for flea control in homes, pets, and livestock creates a demand for pest control services and products. Research. Fleas have been used as model organisms for studying various aspects of parasitism, insect physiology, and host-parasite interactions. In summary, fleas, Siphonoptera, exhibit diagnostic characteristics such as specialized mouthparts, flattened bodies, and strong jumping legs. Examples include the cat flea, dog flea, and human flea. Their parasitic lifestyle, disease transmission potential, impact on pets, livestock, and humans, as well as their role in research, contribute to their economic importance. Effective flea control measures are crucial for maintaining the health of humans, animals, and ecosystems. Plague in detail: causes and vectors, symptoms, cure, etc. Very long answer. ChatGPT. Plague, causes, vectors, symptoms, and treatment. Plague, caused by the bacterium Yersinia pestis, is a deadly infectious disease that has had a significant impact on human history. It is primarily transmitted through fleas that infest rodents. Here's a comprehensive overview of the causes, vectors, symptoms, and treatment of plague. Causes. Bacterium Yersinia pestis. The causative agent of plague is the bacterium Yersinia pestis, which belongs to the Enterobacteriaceae family. Bacterial transmission. Y. pestis is typically transmitted through the bite of infected fleas that have fed on infected rodents. 3 forms. Plague presents in 3 main forms: bubonic, septicemic, and pneumonic. Vectors. Flea-borne transmission. Fleas, particularly species like the oriental rat flea, Xenopsila chiapis, and the human flea, Pullets irritans, serve as vectors by carrying the bacterium from infected rodents to humans. Rodent reservoirs. Rodents, especially rats, are the primary reservoirs of Y. pestis and are critical for maintaining the bacterium in the environment. Symptoms. Bubonic plague. Symptoms: high fever, chills, weakness, swollen and painful lymph nodes, buboes, headache, and fatigue. Transmission. Bubonic plague is transmitted through flea bites or direct contact with infected tissues. Septicemic plague. Symptoms: rapidly progressing fever, chills, weakness, abdominal pain, shock, and internal bleeding. Transmission can arise from untreated bubonic or pneumonic plague or through direct exposure to infected tissues or bodily fluids. Pneumonic plague. Symptoms: fever, cough, shortness of breath, chest pain, rapidly progressing pneumonia, and respiratory failure. Transmission: inhalation of respiratory droplets from infected individuals or animals, leading to person-to-person transmission. Treatment. Antibiotics. Early diagnosis and treatment with antibiotics such as streptomycin, gentamicin, or doxycycline are crucial for a positive outcome. Supportive care. Patients with severe forms may require supportive care, including intravenous fluids, respiratory support, and treatment for shock. Isolation. Patients with pneumonic plague should be isolated to prevent person-to-person transmission. Historical impact. Black Death. The bubonic plague pandemic in the 14th century, known as the Black Death, devastated Europe, leading to the deaths of millions. Other outbreaks. Plague outbreaks have occurred throughout history, impacting various regions and populations. Prevention and control. Vector control. Controlling rodent populations and treating infested areas to minimize flea transmission. Personal protection. Using insect repellent, wearing protective clothing, and avoiding contact with wild rodents can reduce the risk of exposure. Vaccination. Limited vaccines are available for specific at-risk populations. Public health surveillance. Rapid identification and isolation of cases, along with contact tracing, are crucial to prevent outbreaks. In summary, plague is a severe infectious disease caused by Yersinia pestis, primarily transmitted through fleas that infest rodents. Its 3 forms, bubonic, septicemic, and pneumonic, exhibit varying symptoms and modes of transmission. Early diagnosis, antibiotics, supportive care, and public health measures play a vital role in managing and preventing plague outbreaks. Despite advancements in medical knowledge, plague remains a significant public health concern, emphasizing the importance of ongoing surveillance, research, and preparedness to effectively address this deadly disease. 10 diagnostic characteristics and examples, biology, and 10 economic importance of Strepsiptera. ChatGPT. Strepsiptera, twisted-wing parasites. Strepsiptera, commonly known as twisted-wing parasites, is a small and enigmatic insect order characterized by their unique morphology and parasitic lifestyle. Here's a detailed exploration of their diagnostic characteristics, examples, biology, and economic importance. Diagnostic characteristics. Sexual dimorphism. Strepsiptera exhibit extreme sexual dimorphism, with males being winged and free-living, while females are worm-like and entirely parasitic. Reduced forewings. Males have reduced, twisted, and non-functional forewings, halteres, used for balance. Host manipulation. Female strepsipterans are internal parasites, residing within the body of their host insects. Reduced mouthparts. Strepsipterans have reduced or absent mouthparts, as adults do not feed. Complex metamorphosis. Strepsiptera undergo hypermetamorphosis, which includes multiple larval instars with distinct forms. Examples. Xenos vesperum, a strepsipteran parasitizing wasps. Stylops, parasitizing bees. Biology. Parasitic lifestyle. Female strepsipterans attach themselves to the host's body using a specialized structure called a frontal sac, extracting nutrients from the host's blood. Host manipulation. Parasitized hosts often exhibit altered behavior, physiology, and reproductive characteristics due to the presence of the parasite. Male dispersal. Male strepsipterans leave the host to find females, contributing to genetic diversity. Economic importance. Natural regulation. Strepsipterans can help naturally regulate insect populations by parasitizing various insects, including harmful pests. Potential biological control. Studying strepsipterans' parasitic mechanisms could provide insights for developing biological control strategies against pest species. Research. Strepsiptera's unusual biology and complex life cycle make them intriguing subjects for scientific research, contributing to our understanding of host-parasite interactions and evolution. In summary, Strepsiptera, or twisted-wing parasites, possess distinctive characteristics such as sexual dimorphism, reduced wings, and a parasitic lifestyle. They play roles in regulating insect populations and offer potential applications in biological control. The intricate relationship between strepsipterans and their hosts continues to intrigue researchers and contribute to our understanding of insect behavior and parasitic adaptations. Stylopization in detail. Very long answer. ChatGPT. Stylopization, a fascinating parasitic phenomenon. Stylopization is a unique and intriguing parasitic phenomenon that involves the manipulation of the behavior and physiology of certain insects by parasitic organisms called stylopids. This phenomenon is primarily observed in certain species of bees and wasps. Here's a comprehensive exploration of stylopization, including its mechanisms, effects, and significance. Mechanism of stylopization. Stylopids are parasitic insects that belong to the family Stylopidae. The female stylopids are tiny, wingless, and live their entire lives inside the bodies of their host insects. The most well-known example is the genus Stylops, which parasitizes bees and wasps. Infection. Female stylopids find and attach themselves to their host insects. They then burrow into the host's body, where they remain for the majority of their life cycle. Absorption and manipulation. The female stylopids absorb nutrients directly from their host's body fluids, weakening the host's health. Manipulation of behavior. The most striking aspect of stylopization is the manipulation of the host's behavior. As the stylopid larva grows, it sends chemical signals that affect the host's central nervous system, ultimately influencing the host's behavior. Effects on the host. Behavioral alterations. Stylopization can cause dramatic changes in the host's behavior. Infected hosts often exhibit erratic movements, including flying at night, nocturnal behavior for diurnal hosts, and displaying unusual mating behavior. Forced maturation. In some cases, stylopids manipulate the host's reproductive system, causing it to mature faster than normal. This can result in the host engaging in mating behaviors before it's developmentally ready. Death. Eventually, the host becomes so weakened by the parasite's effects that it dies prematurely, often before reproducing. This benefits the parasitic stylopid, as it can emerge and continue its life cycle. Significance and research. Evolutionary relationships. Stylopization offers insights into the complex interactions between parasites and their hosts. The manipulation of behavior suggests coevolution between parasites and their hosts over millions of years. Biological control. Understanding the mechanisms behind stylopization could potentially be harnessed for biological control strategies against pest species. Behavioral ecology. Stylopization provides a fascinating avenue of study for researchers interested in understanding the intricacies of host behavior and parasite adaptations. In summary, stylopization is a remarkable example of parasite-host interactions in the insect world. The manipulation of host behavior and physiology by parasitic stylopids showcases the intricate mechanisms evolved by these parasites to ensure their survival and reproduction. Stylopization not only captivates scientists studying evolutionary biology and behavior but also holds potential implications for pest control and ecological research. 10 diagnostic characteristics and examples, biology and 10 economic importance of Neuroptera. In detail. ChatGPT. Neuroptera, lacewings, antlions, and allies. Neuroptera is an order of insects known for their delicate appearance and intricate wing venation. Lacewings, antlions, and their allies belong to this diverse group. Let's explore the diagnostic characteristics, examples, biology, and economic importance of Neuroptera in detail. Diagnostic characteristics. Membranous wings. Neuropterans have membranous wings with complex venation that gives them a lace-like appearance. Large compound eyes. They typically have large compound eyes that provide good vision. Long filiform antennae. Their antennae are long and thread-like. Chewing mouthparts. Neuropterans possess chewing mouthparts. Complete metamorphosis. They undergo complete metamorphosis, with egg, larval, pupal, and adult stages. Predaceous habit. Many neuropteran larvae are voracious predators, feeding on other insects, aphids, and small invertebrates. Examples. Green lacewings Chrysopode. They are well known for their bright green coloration as adults and their voracious larvae that feed on aphids. Antlions Myrmeleontidae. Antlion larvae create pit traps in sandy soil to capture ants and other small insects. Owlflies Ascalophidae. Owlflies resemble dragonflies but are usually smaller and have distinct antennae. Biology. Larval characteristics. Neuropteran larvae have specialized structures for hunting. Green lacewing larvae bear sickle-shaped mandibles for impaling prey with venomous saliva. Antlion larvae create conical pit traps in sandy habitats. Camouflage. Some lacewing larvae cover themselves with debris to camouflage against predators. Pupal stage. Neuropteran pupae are typically found in silken cocoons. Economic importance. Natural predators. Green lacewing larvae are effective predators of aphids and other small insects, making them valuable for biological pest control in agriculture. Biocontrol agents. Neuropterans, such as lacewings and antlions, are used in integrated pest management programs to reduce the need for chemical pesticides. Pollination. Some neuropterans, like owlflies, can contribute to pollination as they feed on flower nectar. Indicator species. The presence of certain neuropteran species can indicate environmental health, as they are sensitive to pollution and habitat changes. In summary, Neuroptera comprises a diverse group of insects with intricate wing venation, predatory larvae, and various ecological roles. Lacewings, antlions, and their allies play a vital role in natural pest control and contribute to ecosystem health. The delicate beauty and fascinating behaviors of these insects continue to captivate entomologists and enthusiasts alike. Antlions in detail. Importance, structure, food etc. in detail. ChatGPT. Antlions, masters of sand traps. Antlions are fascinating insects belonging to the family Myrmeleontidae, within the order Neuroptera. Known for their unique predatory behavior and distinct habitat preferences, antlions are intriguing creatures with remarkable adaptations. Let's delve into their importance, structure, behavior, and more in detail. Importance. Natural pest control. Antlion larvae are efficient predators, primarily preying on ants and other small insects. They help control pest populations in various ecosystems. Indicator species. Antlion's presence in an area can indicate the health of the ecosystem, as they are sensitive to habitat changes and environmental conditions. Structure. Larvae. The larvae, also known as doodlebugs, have a distinctive body structure. They possess a robust thorax, a large head, strong mandibles, and a conical abdomen. Adult antlions have large, veined wings, a well-developed head, and elongated antennae. They typically have a cryptic coloration to blend into their surroundings. Life cycle. Eggs. Female antlions lay eggs in sandy or loose soil. Larvae. Antlion larvae are notable for their pit-building behavior. They create conical pits in sand or loose soil to trap their prey. Pupal stage. After undergoing several molts, the larva constructs a cocoon using silk and sand particles before entering the pupal stage. Adults. Once pupation is complete, the adult antlion emerges from the cocoon. Predatory behavior. Pit traps. The most famous behavior of antlion larvae is creating pit traps. They dig conical pits in sand and bury themselves at the bottom, waiting for unsuspecting insects to fall in. Ambush predators. When prey falls into the pit, the antlion larva flicks sand to dislodge it, causing the prey to slide down toward the larva. The antlion then captures and immobilizes the prey with its sharp mandibles. Venomous bite. Antlion larvae inject venom into their prey to immobilize and digest them before consumption. Feeding habits. Prey. Antlion larvae primarily feed on small insects, especially ants. They prefer soft-bodied insects that are easier to subdue and consume. Carnivorous diet. The predatory nature of antlion larvae helps keep populations of potential pest species, such as ants, in check. Habitat. Sandy areas. Antlion larvae thrive in sandy or loose soil environments, where they can easily construct their pit traps. Arid regions. They are commonly found in arid and semi-arid regions, such as deserts, where sandy substrates are abundant. Antlions, distinctive pit-building behavior and predatory adaptations make them fascinating subjects for ecological research and a valuable asset for natural pest control. Their role in shaping ecosystems and controlling insect populations underscores their ecological importance. 10 diagnostic characteristics and examples, biology and 10 economic importance of Mecoptera. ChatGPT. Mecoptera, scorpionflies and hanging flies. Mecoptera is an order of insects known for their unique appearance, featuring long and slender bodies with distinctive elongated mouthparts. Scorpionflies and hanging flies are the most well-known members of this group. Let's explore their diagnostic characteristics, examples, biology, and economic importance in detail. Diagnostic characteristics. Long antennae. Mecopterans have long, thread-like antennae that are often longer than their bodies. Mouthparts. Mecoptera have elongated, beak-like mouthparts used for feeding. Wings. They have four membranous wings, with the forewings sometimes resembling those of moths. Reproductive structures. In male scorpionflies, the genitalia resemble the stinger of a scorpion, giving them their name. Examples. Scorpionflies Spinorpody. These insects have long, curving mouthparts resembling a scorpion's tail, although they are harmless. They often feed on decaying organic matter. Hanging flies Bidicidae. Hanging flies have slender bodies and long legs, and they capture prey by hanging upside down and using their elongated front legs to catch insects in midair. Biology. Feeding. Mecopterans primarily feed on a variety of food sources, including nectar, pollen, and small insects. Reproduction. Male scorpionflies often offer a prey item as a nuptial gift to the female during courtship. Courtship behavior. Mecopterans engage in intricate courtship rituals, involving wing displays and movements. Economic importance. Ecological role. Mecopterans, particularly scorpionflies, play a role in decomposition by feeding on decaying organic matter. They aid in recycling nutrients within ecosystems. Pollination. Some mecopterans, such as scorpionflies, visit flowers for nectar and can contribute to pollination. Indicator species. The presence of certain mecopteran species can indicate the health of ecosystems, as they are sensitive to environmental changes. In summary, Mecoptera encompasses a group of insects with distinct characteristics and behaviors. Scorpionflies and hanging flies are intriguing due to their unique mouthparts and reproductive adaptations. While their economic impact is not as pronounced as some other insect groups, they contribute to ecosystem processes and provide valuable insights into the diversity of the insect world. 10 diagnostic characteristics and examples, biology and 10 economic importance of Megaloptera. ChatGPT. Megaloptera, alderflies, dobsonflies, and fish flies. Megaloptera is an order of insects comprising species with aquatic larval stages and terrestrial adult stages. Alderflies, dobsonflies, and fish flies are well-known members of this group. Let's delve into their diagnostic characteristics, examples, biology, and economic importance in detail. Diagnostic characteristics. Aquatic larvae. Megalopteran larvae are aquatic and typically found in freshwater habitats, such as streams and rivers. Elongated bodies. Adults have elongated bodies and long antennae, often resembling a cross between a moth and a dragonfly. Wings. Adults have four membranous wings with a net-like venation pattern. Mouthparts. They possess chewing mouthparts used for feeding on various prey. Examples. Alderflies. Cealidae. Alderfly larvae are aquatic and often found attached to submerged debris. They are voracious predators, feeding on small aquatic insects and crustaceans. Dobsonflies. Corydalidae. Dobsonflies have large, powerful mandibles and are known for their impressive size. Larvae are aquatic predators, while adults primarily feed on nectar. Fish flies. Chaliodinae. Fish flies are named for their aquatic larval habitats. Larvae feed on other aquatic insects and debris, while adults often feed on nectar. Biology. Aquatic larvae. Megalopteran larvae are adapted for aquatic life, with specialized structures for capturing and consuming prey underwater. Pupal stage. Megalopteran pupae are often found attached to submerged surfaces in aquatic habitats before emerging as adults. Feeding habits. Both larvae and adults are predominantly predators, playing a role in controlling populations of aquatic insects and other small organisms. Economic importance. Natural predators. Megalopteran larvae are efficient predators of aquatic insects, helping maintain balanced aquatic ecosystems. Indicators of water quality. The presence of certain Megalopteran species in freshwater habitats can indicate the health of the ecosystem, as they are sensitive to pollution and habitat changes. Research subjects. Megalopterans are studied for their unique life cycles and their role in aquatic food webs. In summary, Megaloptera encompasses a group of insects with distinct aquatic and terrestrial stages. Alderflies, dobsonflies, and fishflies contribute to the ecological balance of freshwater habitats through their predatory habits. Their presence serves as an indicator of water quality and habitat health, making them valuable subjects of ecological research and conservation efforts. 10 diagnostic characteristics and examples, biology and 10 economic importance of Rhaphidioptera. ChatGPT. Rhaphidioptera, snakeflies. Rhaphidioptera is a small order of insects commonly known as snakeflies. These insects exhibit a mix of predatory behaviors and play a role in various ecosystems. Let's explore their diagnostic characteristics, examples, biology, and economic importance in detail. Diagnostic characteristics. Elongated bodies. Snakeflies have elongated bodies with slender abdomens and long wings. Mouthparts. They possess chewing mouthparts, which they use for feeding on various prey. Long antennae. Snakeflies have long, thread-like antennae that are often longer than their bodies. Wings. They have 4 membranous wings with a net-like venation pattern. Examples. Rhaphidia spp. This genus includes various species of snakeflies found in different parts of the world. They are commonly used as examples to study the characteristics and behaviors of Rhaphidioptera. Biology. Predatory behavior. Snakefly larvae are predators, feeding on other insects, including aphids and small arthropods. Life cycle. They undergo complete metamorphosis, with egg, larval, pupal, and adult stages. Egg laying. Female snakeflies lay their eggs on vegetation, and the larvae drop onto the ground when they hatch. Economic importance. Natural pest control. Snakefly larvae are predators of various small insects, including aphids. They help in controlling pest populations in various ecosystems. Biodiversity indicators. The presence of snakeflies in an area can indicate a healthy and balanced ecosystem, as they are sensitive to changes in habitat conditions. Pollination. Some snakefly species feed on flower nectar, potentially contributing to pollination. Research. Snakeflies are of interest to entomologists studying their unique characteristics and behaviors. In summary, Rhaphidioptera, or snakeflies, are a lesser-known but intriguing group of insects. They exhibit predatory behaviors that contribute to pest control and ecosystem health. As indicators of ecosystem balance and subjects of scientific research, snakeflies continue to intrigue entomologists and enthusiasts alike. 10 diagnostic characteristics and examples, biology and 10 economic importance of Trichoptera. ChatGPT. Trichoptera, caddisflies. Trichoptera is an order of aquatic insects commonly known as caddisflies. These insects are well known for their aquatic larvae in diverse habitats. Let's explore their diagnostic characteristics, examples, biology, and economic importance in detail. Diagnostic characteristics. Aquatic larvae. Caddisfly larvae are aquatic and often found in freshwater habitats, such as streams, rivers, and lakes. Protective cases. Larvae construct protective cases using silk and debris found in their environment. Wings. Adults have 4 membranous wings covered in fine hairs that give them a moth-like appearance. Mouthparts. They possess chewing mouthparts, which they use for feeding on plants and detritus. Examples. Phryginia spp. This genus includes various species of caddisflies found in different regions. They are commonly used as examples in entomological studies. Biology. Larval cases. Caddisfly larvae build cases from silk and surrounding materials, which serve as both protection and camouflage. Feeding habits. Larvae are mainly herbivorous or detritivorous, feeding on plant material, algae, and organic matter. Pupal stage. Pupation often occurs within the larval case, which provides protection during this transition. Aquatic habitats. Caddisflies are highly adapted to aquatic environments and play crucial roles in aquatic food webs. Economic importance. Indicator species. Caddisflies are used as indicators of water quality and ecosystem health. Their presence can signify clean and well-maintained freshwater habitats. Ecological balance. Caddisfly larvae help control algae and organic matter in aquatic ecosystems by consuming detritus and plants. Fish food. Caddisfly larvae are an important food source for many fish species, contributing to aquatic food chains. Research subjects. Caddisflies are studied for their ecological role, case-building behaviors, and sensitivity to environmental changes. In summary, Trichoptera, or caddisflies, are remarkable insects with aquatic larvae and various ecological roles. Their presence in freshwater habitats indicates water quality and ecosystem health. As components of aquatic food webs and subjects of ecological research, caddisflies contribute to our understanding of aquatic ecosystems and their inhabitants. External morphology of insects. ChatGPT. The external morphology of insects refers to the study of their physical structure and characteristics visible on the outside of their bodies. Insects display a diverse array of features that have evolved to suit their various ecological roles and habitats. Here are some key aspects of insect external morphology. Body regions. Head contains the sensory organs, mouthparts, and often the compound eyes. Thorax consists of 3 segments, each bearing a pair of legs. In flying insects, it also houses the wings. Abdomen contains the digestive, reproductive, and excretory systems. Exoskeleton cuticle. The exoskeleton is composed of a tough, flexible material called chitin. It provides protection and support for the insect's body. The cuticle is periodically shed and replaced through molting or ecdysis to accommodate growth. Segments and appendages. Insects have a varying number of body segments, typically divided into head, thorax, and abdomen. Appendages include antennae, mouthparts, legs, and wings. Antennae. Antennae are sensory structures located on the head. They vary in shape and size and are used for detecting chemicals, vibrations, and other environmental cues. Mouthparts. Insects possess a diverse array of mouthparts adapted for different feeding habits: chewing, piercing, sucking, sponging, and siphoning. Legs. Insects typically have 6 legs, each consisting of segments and specialized structures for walking, gripping, or digging. Wings. Many insects have wings that are crucial for their mobility. Wings can be membranous or sclerotized and are often adapted for various flight patterns. Eyes. Insects can have compound eyes, which are made up of numerous individual lenses that provide a mosaic view of their surroundings. Some insects also have simple eyes or ocelli, which detect light intensity and direction. Coloration and patterns. Insect coloration varies widely and serves functions such as camouflage, warning signals, and attracting mates. Some insects have aposematic coloration to warn predators of their unpalatability. Respiratory system. Insects use a system of tubes called tracheae to deliver oxygen directly to cells. Reproductive structures. Male and female insects have specific reproductive structures for mating and egg laying. The external morphology of insects is remarkably diverse, reflecting their incredible adaptation to various environments and ecological niches. The study of insect morphology is essential for understanding their behavior, ecology, evolution, and classification. Segmentation and division of the body in detail. Very long answer. ChatGPT. Segmentation and division of the body in insects, known as tagmatization, is a key aspect of their external morphology. This structural arrangement reflects the specialization and functional differentiation of different body regions, contributing to the incredible diversity and adaptability of insects. Here's a detailed overview. 1. Head, cephalon. The head is the anterior-most body region and contains sensory organs, mouthparts, and brain structures. It is highly flexible and allows insects to explore their surroundings. 2. Thorax, thorax. The thorax is the middle body region and is further divided into 3 segments: prothorax, mesothorax, and metathorax. Each thoracic segment bears a pair of legs and, in flying insects, a pair of wings. Prothorax, the first thoracic segment, bearing the first pair of legs. Mesothorax, the middle thoracic segment, usually bearing the first pair of wings. Metathorax, the hindmost thoracic segment, usually bearing the second pair of wings if present and the second pair of legs. 3. Abdomen, abdomen. The abdomen is the posterior-most body region and is typically composed of several segments. Unlike the thorax, abdominal segments do not bear legs or wings. The abdomen houses vital organs such as the digestive, excretory, and reproductive systems. Segmentation. Each body region consists of segments that are often fused, modified, or specialized for specific functions. Segmentation provides flexibility, allowing insects to move and perform various activities. Segments are separated by flexible membranes known as intersegmental membranes. Tagmatization. Tagmatization refers to the grouping of segments into functional units or tagmata. In insects, the 3 tagmata are the head, thorax, and abdomen. The specialization of these tagmata has led to the evolution of different body forms and functions in different insect groups. Division of labor. The division of the body into distinct tagmata allows for the differentiation of structures and functions. The head is responsible for sensory perception and feeding. The thorax is specialized for locomotion and, in flying insects, for flight. The abdomen houses vital systems for reproduction, digestion, and waste elimination. Adaptations. Tagmatization has allowed insects to adapt to various ecological niches and lifestyles. For example, wing modifications in different orders of insects enable various types of flight, while mouthpart specialization suits specific feeding habits. In summary, the segmentation and division of the body into tagmata are fundamental features of insect morphology. This structural arrangement allows insects to perform a wide range of functions, from sensing their environment to locomotion, feeding, and reproduction. Tagmatization reflects the remarkable diversity and adaptability that have made insects one of the most successful and numerous groups of organisms on Earth. General morphology of the head in detail. Very long answer. ChatGPT. The head of an insect is a highly specialized region that houses various sensory organs, mouthparts, and vital structures for feeding, sensing the environment, and communication. Its morphology varies among different insect groups, reflecting their diverse lifestyles and adaptations. Here's a comprehensive overview of the general morphology of the insect head. 1. Cuticle and sutures. The head is covered by a chitinous cuticle that provides protection and support. Sutures, or seams, in the cuticle mark the boundaries between different head regions and segments, allowing flexibility and movement. 2. Compound eyes. Most insects have large compound eyes, composed of numerous individual units called ommatidia. Each ommatidium contains a lens, photoreceptor cells, and pigment cells. Compound eyes provide a mosaic view of the environment, detecting movement and patterns of light. 3. Ocelli, simple eyes. Some insects have 1 to 3 simple eyes called ocelli. Ocelli detect changes in light intensity and direction, aiding in orientation and navigation. 4. Antennae, antennae. Antennae are sensory structures attached to the head and vary in shape, size, and complexity. They are used to detect chemicals, vibrations, temperature changes, and other environmental cues. Antennae play a crucial role in locating food, mates, and suitable habitats. 5. Clypeus and labrum. The clypeus is a part of the front face of the head, often located between the compound eyes. The labrum is a movable plate that covers the mouthparts from above. Together, they assist in manipulating and guiding food into the mouth. 6. Mandibles and maxillae. Mandibles are paired, powerful mouthparts used for cutting and grinding food. Maxillae, also paired, assist in manipulating food and carrying it to the mouth. Mandibles and maxillae are modified according to the insect's feeding habits. 7. Labium and hypopharynx. The labium is the lower lip-like structure that covers and protects the mouthparts. The hypopharynx is a protrusible, tongue-like structure that helps in guiding food into the mouth. 8. Sensory structures. Sensory hairs setae are present on the head and antennae, aiding in tactile perception of the environment. Sensory pits and cincilla, tiny sensory structures, may also be present, detecting chemicals, humidity, and vibrations. 9. Gennae and gula. The gennae are lateral areas of the head between the compound eyes and mouthparts. The gula is a ventral plate that protects the throat region. 10. Genital openings and labial palps. In some insects, genital openings or genital structures are located on the head. Labial palps are paired structures associated with the mouthparts, serving as sensory and tactile organs. The intricate structures of the insect head reflect the diverse functions it serves, from sensing the environment to feeding, communication, and reproduction. The morphology of the head varies greatly across insect orders and species, showcasing the remarkable adaptations that have allowed insects to thrive in a wide range of habitats and niches. Opistognathus head segmentation theory in detail. Very long answer. ChatGPT. The opistognathus head segmentation theory is a concept proposed to explain the arrangement of head segments in insects, particularly in relation to their mouthparts. This theory contrasts with the prognathous and hypognathous arrangements seen in other arthropods. The opistognathous arrangement is observed in many insects and has implications for understanding their evolutionary adaptations. Here's a detailed explanation of the opistognathous head segmentation theory. 1. Background. In arthropods, the head is composed of several fused segments that give rise to different structures such as the antennae, mouthparts, and eyes. The arrangement of these segments in relation to the mouthparts plays a crucial role in understanding the evolutionary history and ecological adaptations of insects. 2. Opistognathous arrangement. In insects with an opistognathous arrangement, the head segments are fused in such a way that the mouthparts are directed backward, beneath the body. This means that the mouthparts are positioned toward the posterior end of the head. This arrangement contrasts with the prognathous arrangement, where the mouthparts project forward, and the hypognathous arrangement, where they are directed downward. 3. Mouthpart position. The opistognathous arrangement results in mouthparts that are positioned closer to the thorax, near the front legs. This arrangement is thought to be an adaptation that allows insects to manipulate and process food more efficiently. 4. Evolutionary significance. The opistognathous arrangement is considered to be a derived character state in insects. It is believed that this arrangement evolved as an adaptation to diverse feeding habits and ecological niches. By positioning the mouthparts closer to the thorax, insects can efficiently process food and interact with their environment. 5. Feeding adaptations. The opistognathous arrangement is associated with a wide range of feeding adaptations. Insects with this arrangement can be herbivorous, carnivorous, scavengers, or have specialized feeding habits. The ability to process food efficiently is essential for survival and reproduction. 6. Examples. Many orders of insects exhibit the opistognathous arrangement, including Coleoptera beetles, Hymenoptera ants, bees, wasps, and Diptera flies. The mouthparts of these insects are adapted for various feeding habits, from chewing and sponging to piercing sucking. 7. Evolutionary implications. The opistognathous arrangement is one of the many adaptations that have contributed to the incredible success and diversity of insects. The ability to feed effectively and exploit various ecological niches has allowed insects to occupy virtually every habitat on Earth. In summary, the opistognathous head segmentation theory explains the arrangement of head segments and mouthparts in insects, with the mouthparts directed backward beneath the body. This arrangement is associated with diverse feeding adaptations and is considered a derived character state that has contributed to the evolutionary success of insects in various ecological niches. Hypognathous theory of head segmentation in detail. Very long answer. ChatGPT. The hypognathous theory of head segmentation is a concept that explains the arrangement of head segments in insects, particularly in relation to the positioning of mouthparts. This theory stands in contrast to the prognathous and opistognathous arrangements. The hypognathous arrangement is observed in some insects and plays a significant role in understanding their evolutionary adaptations. Here's a comprehensive explanation of the hypognathous theory of head segmentation. 1. Background. Insects possess a segmented body plan, and the arrangement of head segments is of particular interest due to its impact on mouthpart positioning and feeding adaptations. The hypognathous theory focuses on the orientation of mouthparts and their relation to the head segments. 2. Hypognathous arrangement. In insects with a hypognathous arrangement, the mouthparts project downward, away from the body. This means that the mouthparts are positioned below the head, directed toward the ground. This arrangement contrasts with the prognathous arrangement, where the mouthparts project forward, and the opistognathous arrangement, where they are directed backward. 3. Mouthpart position. The hypognathous arrangement results in mouthparts that are positioned ventrally, allowing insects to access food sources located beneath them. This orientation has implications for feeding habits and ecological interactions. 4. Evolutionary significance. The hypognathous arrangement is thought to represent an ancestral condition among insects. It is believed that this arrangement reflects the adaptation of primitive insects to their environment and feeding habits. 5. Feeding adaptations. Insects with a hypognathous arrangement may have mouthparts adapted for chewing, sponging, piercing, or lapping. This arrangement is advantageous for insects that feed on substrates located on the ground or on vegetation surfaces below them. 6. Examples. The hypognathous arrangement is observed in some primitive insect orders, such as Diplura and Protura. In these insects, the mouthparts are directed downward, allowing them to feed on organic matter found in soil and leaf litter. 7. Evolutionary implications. The hypognathous arrangement has evolutionary implications, as it reflects the adaptation of early insects to specific feeding habits and ecological niches. While some insect groups have evolved different mouthpart arrangements for specialized feeding, the hypognathous arrangement serves as a reminder of the ancestral condition from which insect diversity emerged. In summary, the hypognathous theory of head segmentation explains the arrangement of head segments and mouthparts in insects, with the mouthparts projecting downward. This arrangement is associated with feeding adaptations that allow insects to access substrates below them. The hypognathous arrangement is particularly observed in primitive insect orders and represents an ancestral state that offers insights into the evolutionary history of insects and their interactions with their environment. Prognathous theory of head segmentation in detail. Very long answer. ChatGPT. The prognathous theory of head segmentation is a concept that describes the arrangement of head segments in insects, particularly in relation to the positioning of mouthparts. This theory stands in contrast to the hypognathous and opistognathous arrangements. The prognathous arrangement is observed in certain insect groups and plays a significant role in understanding their feeding adaptations and ecological interactions. Here's a comprehensive explanation of the prognathous theory of head segmentation. 1. Background. The arrangement of head segments and mouthparts in insects is crucial for understanding their feeding habits, sensory perception, and ecological roles. The prognathous theory focuses on the orientation of mouthparts and their relation to the head segments. 2. Prognathous arrangement. In insects with a prognathous arrangement, the mouthparts project forward, in front of the head. This means that the mouthparts are positioned anteriorly, allowing insects to reach out and feed on food sources located in front of them. This arrangement contrasts with the hypognathous arrangement, where the mouthparts project downward, and the opistognathous arrangement, where they are directed backward. 3. Mouthpart position. The prognathous arrangement results in mouthparts that are positioned at the front of the head. This orientation has implications for feeding adaptations, sensory perception, and interactions with the environment. 4. Evolutionary significance. The prognathous arrangement is believed to represent an ancestral condition among insects. Early insects likely possessed this arrangement as they adapted to feed on materials located in front of them. 5. Feeding adaptations. Insects with a prognathous arrangement may have mouthparts adapted for biting, chewing, or lapping. This arrangement is advantageous for insects that feed on materials found on surfaces in front of them, such as plant tissues, nectar, or other insects. 6. Examples. The prognathous arrangement is observed in various insect groups, including Coleoptera beetles and Hemiptera true bugs. These insects have mouthparts that are well suited for biting and piercing sucking, respectively. 7. Ecological implications. The prognathous arrangement has ecological implications as it reflects the adaptation of insects to specific feeding niches. Insects with this arrangement are often involved in diverse ecological roles, including herbivora, predation, scavenging, and parasitism. 8. Evolutionary implications. The prognathous arrangement provides insights into the evolutionary history of insects and their adaptive radiation into different ecological niches. While some insect groups have evolved alternative mouthpart arrangements for specialized feeding, the prognathous arrangement is a reminder of the ancestral condition that has contributed to insect diversity. In summary, the prognathous theory of head segmentation explains the arrangement of head segments and mouthparts in insects, with the mouthparts projecting forward. This arrangement is associated with feeding adaptations and sensory perception that allow insects to interact with food sources in front of them. The prognathous arrangement represents an ancestral state that offers insights into the evolutionary origins of insects and their ecological roles. Head segmentation theories about the segmentation of the head in detail. Very long answer. ChatGPT. The segmentation of the head in insects is a complex topic that has intrigued entomologists for centuries. The head is a critical region of the insect body, housing sensory organs, feeding structures, and other vital components. Theories about head segmentation have evolved over time as researchers have studied the diverse arrangements of head segments and their implications for insect morphology and evolution. Here's a detailed exploration of some of the key theories about head segmentation in insects. 1. Tripartite theory. One of the earliest theories proposed by scientists like Dorn and Wheeler in the late 19th and early 20th centuries suggested a tripartite arrangement of the insect head. This theory proposed that the insect head consists of three primary segments: the protocephalon forehead, detocephalon midhead, and tritocephalon hindhead. Each of these segments was believed to contribute to specific structures, including sensory organs and mouthparts. 2. Tagmata theory. The tagmata theory is based on the concept of tagmosis, where segments fuse together to form functional units called tagmata. According to this theory, the insect head is composed of several segments that have fused to form distinct tagmata, each contributing to specific functions. For example, the antennal segment, intercalary segment, and nathal segments are thought to contribute to the head's structure and sensory capabilities. 3. Paired segment theory. Some theories suggest that the head is composed of paired segments, with each pair representing specific functions. For instance, the paired theory posits that the first segment is paired with the second, the third with the fourth, and so on. Each pair is believed to contribute to specific head structures, such as mouthparts and sensory organs. 4. Evolutionary modifications theory. This theory focuses on the idea that head segmentation has undergone various evolutionary modifications over time. Insects have adapted to diverse ecological niches, leading to changes in the arrangement and fusion of head segments. Different insect groups exhibit various patterns of head segmentation based on their feeding habits, locomotion, and sensory requirements. 5. Adaptive function theory. Another perspective suggests that the arrangement of head segments is adapted to specific ecological functions. For example, insects with mouthparts suited for piercing sucking may have different head segment arrangements than those with chewing mouthparts. The arrangement of sensory structures like antennae may also be influenced by the insect's habitat and lifestyle. 6. Integration of gene expression. Modern genetic studies have provided insights into the molecular mechanisms underlying head development. Gene expression patterns play a crucial role in determining the formation and differentiation of head segments. The integration of genetic processes, along with interactions between signaling pathways, contributes to the establishment of distinct head structures. In summary, the theories about head segmentation in insects are diverse and have evolved over time as our understanding of insect morphology and development has advanced. These theories range from early concepts of tripartite arrangements to more modern insights into genetic processes and